All right, good morning, everybody. My name is Gabriel Pagnotti. I'm a postdoc in the Department of Biomedical Engineering here on campus. I want to share with you a body of research that our lab has been conducting over the past few years, utilizing a novel strategy, mechanical signals, as a means to combat cancer-induced bone loss. So I think a general consensus among everybody in the audience is that cancer is a very debilitating disorder, but I think what we don't recognize is the secondary effects that occur to the musculoskeletal system. So here we have an example, cancer cells. They attack the bone marrow and they cause this kind of Swiss cheese effect in this forearm, this person's forearm, removing bone mass and ultimately increasing fragility. We go over here to another example where there's a spinal vertebrae. These cancer cells have invaded the bone and ultimately eaten it from the inside out. This is actually a spinal compression fracture, which will ultimately lead to an issue of paralysis. So we have cancer treatments, right? Chemotherapies, radiotherapies, and for the most part, these have become pretty effective in recent year in challenging the tumor burden or the cancer as a whole. But what happens is that when we come out of this, what you can't outpace is the secondary effects, the amount of bone loss that occurs over the treatment or over the progression of the disease itself. So I'm talking about bone, and like, why do we care about bone? It's not a big deal, we just beat out cancer, right? Well, the reason it's so important is that as we decrease our bone mass, there's an inverse correlation between decreasing bone mass and increasing fracture risk. So over time, this increase in fracture risk ultimately decreases our quality of life, and this can increase mortality rates. So in order to propose new strategies to combat this bone loss, we need to understand how bone works in general. It's very active tissue. It readily degrades, breaks down, and builds back up again over time. This is a natural process called bone remodeling. And it's sort of this like Zen-like situation where whatever you take out, you kind of have to put back in, and that helps keep bone healthy over time. So take, for example, exercise. Physical activity. We just had the US Open, and encouraging bone to grow even more. So a pro-formative effect, exercise, the same situation here with Venus Williams, and her arms are about the size of my head. And that's not by coincidence. We have a mechanical signal that's generated by a ball striking the bracket, and if you could imagine a signal propagating up her arm and into her bone. Not hard to imagine. We know that exercise encourages musculoskeletal health. So actually her dominant arm is roughly 30% more dense than her non-dominant arm. Now, if we want to exemplify how dependent bone is on mechanical signals, we can remove them entirely. So somebody at chronic bed rest, such as this person, is going to experience very quickly musculoskeletal atrophy. You throw cancer into the mix, and now you've accelerated the situation. So it really is a point of concern. Now, in order to combat this type of bone loss, obviously we want to look for something outside of a pharmacological agent or chemotherapy. So we look back at exercise. It seems like a pretty promising strategy to combat bone loss, but as you can imagine, somebody with very weak bones is not going to get out there and start jumping hurdles or lifting 1,000 pounds like this guy. So, and that really derives from the intensity of the signal overall, right? The bone is very weak. Subjecting them to a signal or a mechanical signal of this magnitude is probably going to induce the fracture it was intended to prevent in the first place. So we have to come up with another strategy, but still keeping in mind the mechanical signals, which are so beneficial to maintaining bone health, we'd like to incorporate that into this new strategy. So just something a little bit more subtle. So more of it, just in smaller amounts. So low-intensity vibrations, which I'll kind of refer to as LIV from now on, is a treatment strategy that our lab and others have demonstrated as very beneficial, very osteogenic, which means it helps bone grow in situations of osteoporosis. So essentially what happens is somebody stands on this vibrating platform, they get jogged up and down, very subtle signal, something reminiscent of what this chart here on the side. The signal propagates from the heel, up the hind limb, up the leg, across the spine, and terminates somewhere around the base of the skull. Now we know that this is very beneficial in terms of osteoporosis. So can it be used as a strategy to combat cancer-induced osteoporosis? Maybe. So in order to answer this, we have two different models of disease, two different questions that we need to have answered. One is safety, and the other one is efficacy. So if it's safe, good. Then we can apply it and see if it actually works. So in our first model, we have a genetically susceptible model to ovarian cancer. And the idea here is that we want to expose these animals to this signal, this very subtle signal, and see if it teases out the cancer itself, right? It makes no sense if we're building bone, if the animal is actually creating more tumorigenic tissue in the process. So if that works, we move on to another animal model, multiple myeloma, which is a very aggressive bone marrow cancer. It literally eats the bone from the inside out very rapidly. So in looking at these two models, we can actually answer whether or not this may be an effective treatment strategy to combat cancer-induced bone loss in the future. Okay, looking at our first animal model, right? This is the ovarian cancer model, and we're determining whether or not the LIV signal will encourage tumor growth or accelerate the death of these animals over time. So we tracked them. We have a healthy cohort. We have our untreated group and those that were treated with LIV. Both the untreated and the LIV treated group are susceptible to developing the cancer. So we followed these animals over the course of 1 year, and what we found out was that they have identical tumor burdens and that there was no significant deviation between the two groups tracked over this 1 year's time. So it seems to be a pretty safe strategy at this point. We're a bone engineering lab, so we like to quantify stuff, and this is one of our favorite assays, is running micro-CT. It gives us a nice idea of the amount of bone that we have. Here we have a femur, so we've quantified the healthy bone, or the healthy cohort, and in comparison to our untreated group, we have about almost a 50% decrease in bone loss over this 1 year period of time. Compare that to our LIV treated group, and we've suddenly saved about half of the bone, and that seems pretty critical, but we want to test this in our model that's explicit to bone cancers. So we move to our model for multiple myeloma. Again, we have a healthy cohort. We have two models. Both have been injected with myeloma cells, so 100% of the population has acquired the disease. However, one is untreated, and the other one, again, treated with LIV. We tracked these animals over 8 weeks. And again, this is a very aggressive cancer. So if we were going to see it, we're going to see it pretty quickly. We looked, again, using micro-CT. We see some pretty healthy bone in our first cohort. And then looking at the untreated group, we have almost a 75% decrease in bone, which is, we see the bone just plummet. And this is over the course of 8 weeks. Then we compare this to our LIV treated group, and we've saved roughly half of that same amount. So at this point, we can say that there is some element of clinical or preclinical efficacy in being able to, A, safely administer these signals to somebody susceptible to the disease, and we're able to save bone at the same time. Okay, well, this is fine for mice, but we'd like to see this translated into human. Our colleagues over at St. Jude's recently performed a clinical trial on childhood cancer survivors. We have a 60 cohort or 60 subject study that was performed over the course of 1 year. 30 were given a placebo plate and 30 were given an LIV plate. So over the course of 1 year, what we can see is an actual drop from baseline in our placebo group. This is the bone mineral density. And then in the LIV tree group, we actually have a significant amount that was increased from baseline. So it seems to be at this point, not only do we have preclinical evidence that it's safe and effective, but we actually have this translated into the clinic. And we can see that it combats long-term pediatric cancer-induced osteoporosis. So what we'd like to do in the future is apply this to other forms of bone cancer, like breast cancer metastases, these readily home to bone, and virtually eat it out, eat from the inside out. So now we know that it works, we want to know how it works. So we're currently running studies to clear that up. I want to thank everybody, including the mice that donated themselves for these studies, everybody in my lab, including my PI, Clint Rubin, who's chuckling in the background now, and everybody else who contributed to these studies over time. I think it was worth the effort. Thanks.